My name is Greg Cosgrove. I'm the Chief Medical Officer for the Pulmonary Fibrosis Foundation. Hard to follow a discussion like that. Hope. One of the key things that we all strive to have. It provides a path forward, and that's what I took home from this message here. We have hope. Not only that, we have individuals with whom to work and to fight, as Dot said. I've characterized her, Dot Del Rosso, who is that? She's a firecracker. She just proved it again. So I would encourage all of you to talk to each other. The disease that we're here to try to fight affects each and every one of us in different ways. But if you ask the person next to you, the person at work, a friend that you meet at the shopping center, you'll find more people affected by the disease. And we have more people to join us in the fight. So I'd encourage you to do so that we do not utter the words, "What is pulmonary fibrosis?" anymore. So I'm pleased to continue this morning's program, and we have an esteemed keynote speaker with us today, who I think will be able to demonstrate the impact of how we can care for patients with complex disease, and that is Dr. Edith Perez. But I'd like to turn over to my colleague, Dr. Christine Garcia, to do a more formal introduction. So on behalf of everyone here, the Foundation, I'd like to welcome you to the PF Summit.
Thanks, Greg. I have the pleasure of introducing our keynote speaker. Dr. Edith Perez is a professor of medicine at the Mayo Clinic. She is the director of Mayo Clinic's Breast Cancer Translational Genomics program. Her experiences include leadership in academics as well as the pharmaceutical environments, and she has an added focus on philanthropic endeavors. Please welcome Dr. Edith Perez.
Good morning. Do we have an advancer for the slides, or should I just? We're going to do some magic here together, it seems. Okay. Okay, someone's running. That's a good sign.
I'll get it.
Okay. Thank you. Thank you very much. Talk about preparation for a big meeting like this one. Truly, really a pleasure for me to be here today. I've been here since yesterday, and I can tell you I learned so much from participating at the poster session yesterday. I actually talked to many people who were gracious enough to talk with me, and the learnings were quite impressive. And before I show you the next slide, we in the oncology community have so much to learn from you, and I certainly hope that in the panel session we can expand on this a little bit. Some disclosures: the views are certainly on my own. I prepared the slides, and I would like to see that you also know that I am completely honored and privileged to be here today. And in that same vein, I'm also very respectful of your time. So I hope that the next 30 minutes will provide you with an overview that will address how we develop drugs in the oncology space, how things have changed over the years, as well as consider how these learnings can be applied to each one of your roles as part of PFF or pulmonary fibrosis or academia, industry, wherever you sit, that you will get something out of my talk. So I would like to see the next slide. Oh, I can see only myself here, and the slides are there. So this is interesting. Stress test, okay. I usually have a blood pressure of about 110/70. Okay, here we go. Wow. So I decided, since I was invited to give this talk, I actually have read so much about pulmonary fibrosis. As some of you mentioned earlier, not too many people in the general community talk about pulmonary fibrosis, but a member of my family had pulmonary fibrosis, which touched me tremendously. But since being invited, I think I know a lot more about the disease than the usual hematologist, oncologist. So I decided to put this one slide to frame where I see individualized patient management in idiopathic pulmonary fibrosis as a benchmark for the rest of the conversation this morning. Certainly, there are non-pharmacologic options for management that certainly include lung transplants for some patients. When we go to the right part of the slide, there are pharmacologic options. And I was actually almost shocked to realize that there are only 2 FDA-approved therapies for pulmonary fibrosis, both of them approved the same day in 2014. So there's a lot of work ahead. First of all, to not only optimize learning so that patients get access to these medications to delay progression of the disease, but also to consider new approaches that can be taken through the conduct of clinical trials, participation in clinical trials, new science, as well as collaboration. So here's an outline of what I will discuss with you this morning in more detail. I'll spend actually the majority of my time discussing principles of innovative drug development, and I'll have about 19 slides to address this topic. Then I will have about 10 slides or so to discuss regulatory strategies for expedited drug development, the potential role of real-world data as well as patient-related or reported outcomes, and then I will close with a few summary slides. When I think about 21st century oncology trials, we must stress that everything is centered on patients. Everything we do in oncology is centered on patients, and that includes really advocates for patients. These trials all involved a collaboration with clinicians, translational scientists, funding agencies, biopharma, as well as institutional support. What are the data gaps that we must address as we develop innovative translational research? First, which patients benefit the most? Why is it that some patients don't respond to the therapies that we administer? How should molecular profile or understanding of the biology of cancers or diseases influence trials as well as therapy? If we identify a biomarker that is relevant to a particular disease that a patient suffers, what is the prevalence of that marker that will help us figure out how long it may take to accrue patients to a clinical trial? How do we optimize sequencing treatments? And can we develop clinical data to support technology development so that we can have surrogate endpoints instead of the traditional endpoints that may take a year or 2 years to realize that may in turn lead to drug approvals? But in addition to thinking about data gaps, we also have to think about external challenges that we need to address as we conduct research. What is the regulatory landscape? What is the competitive environment? What are the expectations of patients as well as regulators? How complex is the disease? Because sometimes just using one drug will not fix it all. There's also a rapid evolution of technology as well as increasing payer and Medicare pressure. Again, all of these things are important for all of us to piece together as we look towards the future. So there are several things that we consider every time we develop a clinical trial in oncology. And first is the patient population. What is the cancer subtype? And these days, the cancer subtype determination is based on histology as well as biomarkers. I will talk with you a lot more about how we view biomarkers in this space because I think it's totally applicable to what's happening in pulmonary fibrosis. We also need to think about the prevalence of the disease as well as the current standard of care with outcomes. Because if we have a new approach, we have to think about how is this new approach going to fit in with what we know works or doesn't work as well as we would like. Certainly, we have to think about the potential impact and significance of the results of the trial if successful. We have to think of what we're going to do next if the trial is successful. As important to anything else is the feasibility, because sometimes we sit in our offices or our laboratories and we create these massive trials, very complicated, and unless we involve patients and patient advocates as we develop these trials, the trials may not be as feasible as they should be. so that they can be properly activated and conducted. Because it's more than having an idea. It's actually putting an action plan so that the idea can come to fruition on behalf of our patients. So a lot is happening with cancer subtyping, and I really hope that this is going to be the future for idiopathic pulmonary fibrosis. In oncology, tumor sequences is becoming actually standard in clinical practice. Liquid biopsy technology is being adopted. So in the past, what happened in oncology is that we needed to obtain big biopsies. We needed to actually put the tumor specimens in frozen containers. So it was hard for some patients to undergo initial biopsy or even repeated biopsies as we conducted clinical trials or clinical practice. So there's a big emphasis for developing liquid-based technologies in which we, by just doing a blood test, we can analyze DNA and protein changes that may indicate whether a patient may be eligible to receive therapy, but also may indicate whether the therapy is working or not working even before we see any radiographic or symptomatic changes. We believe that right now we may call lung cancer, but in the years to come, as an example that I'm using of lung cancer, we will have, and actually it's happening today, we will have EGFR lung cancer, ALK-positive lung cancer, PD-L1 positive lung cancer. So things are really rapidly evolving in our field, and I really hope, again, it's going to also happen in your field. Certainly, as we think of the evolution of utilizing biomarkers to better define cancers, clinical trials have to increasingly use this novel sequencing technology and enrollment, because we want to utilize the best biology to develop the best clinical trials. And the more we do in this arena, the more there will be earlier intervention and adoption of incorporating molecular tools to subdivide cancers for best patient management. This includes clinical trials, as well as the attention that health technology agencies and regulatory agencies, such as the FDA in the United States, EMA in Europe, or regulatory agencies around the world, will think, okay, in order to approve new drugs, we're going to look at the subtype of patients who may benefit the most for these new drugs, approve them readily, and not only approve them readily, but allow patients to have access to these new treatments. There's many criteria that we utilize when we think about what agents to test. Well, the agents need to have relevant mechanism of action. There has to be some scientific rationale. We need to have some preclinical in vitro data in the laboratories. We need to assess whether the agents may be tolerable based on what we call initial phase 1 clinical trials. We need to have some understanding of how the body metabolizes the agents, tolerability. And increasingly, we need to define whether we have some biomarker that, again, can identify whether one therapy has more likelihood of benefiting a patient versus another therapy. Certainly, as we think of new agents, and this was exemplified by many of the presentations at the posters yesterday, we need to think about first in class or best in class, because we want things to be better for our patients. So what are we doing in oncology? Again, that may be totally applicable to pulmonary fibrosis. The historic approach had been that we have a study arm and we have a control arm of patients, and we did large trials in unselected populations. So we took, again, I'm going to use the example of lung cancer. We took large numbers of patients with lung cancer, and we tested drug A versus drug B. And what's happening now is that we're taking this novel approach in which actually we are subdividing these lung cancers in many different subgroups. We're starting to do more adaptive and smaller trials so that we can have earlier and speedier development of new treatments for specific subtypes of patients in the field. So as part of this new approach, we're also integrating this earlier with later clinical trials. And let me give you an example using this particular slide and the cartoons. In the past, we used to do what we call phase 1 clinical trials, and these are trials in which we typically evaluate increasing doses of drugs to determine what is tolerable. And then we would do phase 2 clinical trials in which we would say, again, I'm going to use the example of lung cancer in this case, we would take hundreds of patients to say, okay, this drug was efficacious, so was safe in a phase 1. Let's see if it's going to be efficacious in phase 2. And then if we show that efficacy, then when we conduct comparative trials that would involve thousands of patients. Well, we're changing that dramatically because this is just not good enough for our patients. So what we're doing these days is that we're trying to identify biomarkers really early. So even in phase 1 trials, when we're looking at safety of a drug initially, we're trying to ask patients who are most likely to benefit from the very beginning to participate in these initial trials. And then we expand the phase 2 trials. So you may wonder, well, this sounds very interesting, at least it's interesting to me, but how is this translating to faster development of drugs that are approved by regulatory agencies, such as the FDA? So this gives you an example of how this is translated to reality for our patients. In the past, as we're conducting this phase 1, phase 2, and then phase 3 clinical trials, it was taking 8 to 10 years until we got the results of these phase 3 trials to ultimately lead to an FDA approval. And with this new approach in which we are looking at histological subtypes, molecular subtypes, and doing multiple genomic subsets of trials, sometimes we're getting approvals by the FDA after phase 1 clinical trials. So the timing from development from the laboratory to the patients is being significantly shortened by thinking of a biomarker-driven strategy for drug development. And the idea here is to reduce time and cost of developing promising drugs, taking into account the heterogeneity of cancer, as we want to conduct more efficient trials with innovative designs. So I've mentioned biomarkers quite a bit. So let me deal with this a little bit more for all of us this morning, because again, it's so applicable to what I saw yesterday at the posters. Co-development of reliable biomarkers is critical to facilitate development of effective therapies. And we also want to identify patients who may have good prognostic signatures for whom aggressive therapies are not needed. Because it's not one treatment for all. We need to understand who's likely to have disease progression quicker or slower so that we can utilize the right approaches for clinical trials as well as treatment in the clinics. So certainly, as we identify biomarkers, we need to figure out that the tests are effective and validated, as well as meet standards that can be used in the product labels. So this slide is busy, but one of the things I neglected to say is that every time I present slides, my slides are totally available to everybody. So certainly you can send me an email, or I'm sure the PFF will be happy to send you the slides so that you can take a little bit of extra time going over the material, as well as potentially use the materials in educational activities with others. But as we developed companion diagnostics for targeted therapies, and in oncology we call these omics-based biomarker tests. So it can be proteomics, metabolomics, genomics, okay? So what we need to think is about how do we handle the specimens? What kind of testing do we need to do? Then we need to determine the clinical utility of a biomarker that is developed, as well as how to interpret the results obtained. This is really, really important as we think about current therapies, current patient management, as well as the future. And in cancer, this is a tremendously relevant situation, and this may happen again in pulmonary fibrosis, but in cancer, the problem that we have, first of all, people are getting cancer. That's big enough of a problem. Number two, in the United States, more than 2 million persons are diagnosed with cancer every year. Globally, it's more than 20 million every year. But what happens in cancers is that there are many molecular changes in cancers, many mutations, even when the tumors are diagnosed early. But as time progresses, there could be hundreds of alterations that we need to think about how to prioritize in terms of treatment as well as study development. So we always need a systematic approach on how to evaluate all these changes, and having the strategies to do appropriate biomarkers is really critical for the future. So as I think about biomarkers, it's more than pie in the sky when we do clinical trials. Because I can do a clinical trial, and I can do whole exome sequencing, I can do single-cell RNA, whatever test you can think of. But when I write a trial, I need to be really specific on how I would like things, or how our team wants things to be done, so that we can get a good set of specimens that can help the science as well as patients. We need to tell people exactly how to prepare the blood, how it should be stored, how it should be shipped. We need to have systems for sharing some testing data with investigator and the patients. Again, this is something that is changing a lot in oncology. Because in the past, the way we used to do trials, we would ask patients, they donate your specimen, and let us do what we need to do. Sometimes we would say, not only give us one specimen, how about if you give us another specimen in 2 months? And people would think, okay, I want to do something for myself, I want to do something for my relatives, I want to do something for the future. But the way we're thinking about this is that if we're asking the patients to participate in trials and donate some of their specimens, I want the patients to get something back. I want the patients to get some of the information that we get from the biomarkers, because that can help in 3 aspects. Number 1, engagement of patients. Number 2, it can help the patients understand what's happening with the disease. And number 3, it may inform the patient as well as the clinicians of potentially new trials that the patients may be involved in if they have that biomarker information. So again, as a theme of my presentation today, you're seeing that there was a way we used to do things, and there's a new way that we're looking at things so that we can expedite the understanding of biology as well as development of new treatments for patients. As important as anything is to have patient education materials. Because I tell you, we as scientists can have all this terminology, all these acronyms, and we need to be sure that the information that we want to share with patients as we conduct trials is digestible for the patients who will participate in the trials, for the patients who will help us talk to others related to participation in these studies. I'm actually going to use an example of what we're doing in our laboratory to deal with this complexity of biomarker testing in cancer. So I'm going to show only 3 slides of the work in our laboratory, but hopefully it will inspire you related to what's really happening today and how we view what's happening today as a tool to make things better for the future. So essentially what you have in the middle of this slide could be a picture of a slide from a biopsy. And essentially there are 5 different profiling modalities that we can take to interrogate tissue samples in oncology. We can do geometric profiling, as we can take different areas of the tumor and just do circles or squares or little diagonal things. We can do some segment profiling by looking at the different cells that are seen in the histological specimen. We can do some what we call contour profiling, looking at what are the cells next to, because that may be relevant for understanding what's happening in the tumor cells. We can do gridded profiling, in which we can do we can look at everything, or we can do some rare cell profiling. So let's say we're interested in a cell type, and we are then looking, okay, where can we find that cell of interest that we're looking at in the context of a tumor? So it's always good to have kind of a general way to conceptualize what we do. But in addition to that, let's put this into the test. So here we have another picture of a tumor slide. And by ROI, we define that as regions of interest within a tumor. So what we do is when we take a tumor specimen, we look at it under the microscope, and then we segment the tumor into different areas to help us understand what's really happening in the tumor. So we actually select these regions of interest, and then we select for tumor, and that's seen in green. CD45, which actually stands for lymphocytes, and CD68 in red are actually the macrophages. And certainly in cancer, all of these components are tremendously important. Certainly pulmonary fibrosis is tremendously important to understand what's happening with the immune system, what's happening with the lymphocytes, what's happening with the macrophages. Where are they? Are they there at all? Or can we figure out where they are, what they do, as well as potentially increasing the migration of these important cells so the tumors can be killed, okay? So this is completely new technology that just became commercially available last week. However, we've had this technology in our laboratory for the last year and a half. So we've been generating data, and we at Mayo and our laboratory in Jacksonville, we're very proud of this. We were the first one who received the commercial machine, and it's already been tested as part of our clinical trials. But you may say, oh my gosh, this sounds kind of interesting, nice pictures, thinking about the tumor, thinking about the lymphocytes, because remember, we need the lymphocytes to be there to kill tumors as well as macrophages. But there's so many types of lymphocytes, there are so many different cells that are potentially important for the immune system, which is where I'm focusing a lot of our interest right now. How can you do this with one slide? Because that's what we're trying to get at. We cannot get 800 slides to test 800 proteins, or 40 slides to test 40 proteins. So the way we're doing this is highlighted on this one slide. So this machine is called the GeoMX. DSP stands for Digital Spatial Profiling, in which we can do high-plex protein and RNA profiling in tumor specimens. So what we do is that we have protein reagents, we have RNA, which as you know come from DNA, and then we have these probes, and then we put one slide of tumor into this machine with all of these probes. And with this new technology, we can do, today, we are doing 40 different proteins involved in the immune system with one slide. So then what we do with this is that we looked at all of these different regions of interest within the tumors, and then the machine actually tells us the expression of 40 different immune-related proteins in each one of these areas of the tumor. And by doing this, we're really subcategorizing what's really happening in the tumors way more than what we could do by just looking at one slide under the microscope to determine whether someone had breast cancer, gastric cancer, or lung cancer. And to me, this is really the new era of what's happening in biomarker development in oncology. Certainly, we're looking at genes, we're looking at proteins, and we're looking at the localization of immune proteins in the context of the tumors, the stroma, to determine how we can maximize the ability of the immune system to control cancer. And just talking about this makes me excited about where we are and the potential future. So as we think about these novel technologies, we need to address low prevalence, and this is an issue for your community. There's some mutations or molecular alterations that are important and what we call drivers, and they're just kind of passenger mutations. We just happen to be there, but they don't have really an impact on tumor growth or invasion. Tumors are different. Some agents work a little bit, and so we need to think about putting them together. So in oncology, as is probably happening in your field, we need to think about not only about developing single drugs, but how to combine drugs. Luckily, the FDA has guidance of co-development of 2 or more agents, and the reference is right here. So it's good that the FDA is thinking with us about how to think about better strategies. As we do trials, we need to think of recruitment. What are the challenges? But what are the solutions? In terms of challenges, everybody diagnosed with cancer is totally scared, and I tell you, respect that. All of you, healthy or persons diagnosed with pulmonary fibrosis, all of you women go and have your mammograms, classically. Men worry about, do they have the PSA for the potential of identification of prostate cancer, and I can used to other things, HPV testing, all of that. We're all very sensitive human beings, and we should be actually. So when someone has cancer and we approach a patient related to talking about clinical trials, we need to be sensitive to the emotional condition of the patient when that person comes to us. We need to think about what we're asking of patients, but we're also thinking to solve this, we need great people conducting clinical trials with us. We need to be able to share with patients, my gosh, this is an opportunity, and think about taking part of this opportunity to look towards better outcomes for you as well as the future. So I've talked a lot about the principle of innovative drug development. What are we doing differently in oncology in terms of the design of clinical trials? How are we thinking of what agents to test? How are we thinking about combinations of new agents? How are we thinking about the development of biomarkers to subcategorize cancer? So in the last few minutes, let me address these 3 other important areas. we need to develop innovative endpoints for drug development that are relevant, so we want to collaborate. We want to optimize these approvals, and the FDA has come up with really some very interesting strategies to expedite approvals, and they're called priority review, breakthrough therapy, accelerated approval, fast track designation, as well as orphan drug designation. We won't have time to go over the details of each one, but I wanted to just show this one slide with you. And it is that this timeline for introduction of expedited approval pathways is occurring in the United States. You see the flag of the United States, the European Medicines Agency, as well as the Japanese group through PRIME. So through Sajigaki, excuse me, PRIME is for Europe. So essentially, there's a global thinking of expediting drug development. So here's just one slide that has some more detailed information on the differences between breakthrough therapy, accelerated approval, priority review. And again, this will be available to you. But the bottom line is that all of these classifications lead to incentives to manufacturers that include fee waivers, market exclusivity, tax credit, research grants, again, with the idea that time to market is crucial for patients as well as manufacturers. Real-world data. And I saw a great poster yesterday from one of my colleagues at Mayo addressing real-world data and real-world evidence, because we need to improve the efficiency of clinical research by capitalizing on data that are captured every day, beyond the clinical trials that we do. So I'm glad that you have a patient registry, because it will help with real-world data and analysis. We need, obviously, all of us as physicians to be clear and consistent on our documentation so that the real-world data can actually be used in the proper way. There's also the era of big data in oncology and other diseases with wearables and direct data reporting by patients that we need to take advantage of. Many fancy things. Electronic patient-reported outcomes. This is another complete change, where instead of the patients just coming once every 3 weeks for follow-up to the clinic, the patients actually are reporting how they feel on a daily basis through electronic ways of communicating with their investigators as well as physician treatments. So as I'm closing with actually 3 slides, the core focus that we have in bioncology is a continuous learning of basic research, clinical research, scientific understanding, academia, development, medical affairs, biomarkers, with a scientific focus on operational and team effectiveness. I think there's a new paradigm for oncology and pulmonary fibrosis trials. Increased patient screening and participation into trials, global collaboration, innovation to provide highly effective agents faster to our patients. Access meaningful data at scale, get clinical insights, and realize the value from our insights to develop personalized translational therapies. On this note, I hope that these 35 minutes provided you with the view of what's happened in the past, how we view what's happening today, and how we view the future. I thank you, and anything I do, anything I've done in my life is because I love people, I love life, I love the opportunities that we all have, and I have great collaborators. And this happens at Mayo Clinic, funding from the Komen Foundation, Stand Up to Cancer, the 26.2 with Donna Foundation, and I love to run and walk. And I want to also thank many friends that I have, including a tremendous friend of mine who's on the audience, Christian Ascarati. Thank you very much, guys.